Hello, everyone. Thank you so much for joining our webinar today, Testing of Contaminants and Adulterants in Dietary Supplements, a risk-based approach. I'm very excited to have Lukas Vaclavik and Stassi here with me today to host the webinar. I am Ellie Abraham, and I will be moderating today's session. Today's presentation will be about 45 minutes. Before we begin, I'll let you know more about how this webinar will run. The webinar is being recorded, and the slides and recording will be available for you within 3 business days. A short Q&A session will follow the presentation to answer viewer-submitted questions. During the webinar, you can submit questions you have using the webinar sidebar menu. Select the Questions tab, type in your question, and then hit Enter on your keyboard. Remember, you can submit questions throughout the webinar. That concludes my brief introduction today. Jeff, I will now pass the webinar off to you for our introduction. You may begin your presentation.
Thank you, Ellie. And again, thanks everybody. We appreciate your attendance today. And Lukas and I have a very interesting topic, I believe, and a very timely topic. We're going to talk a bit about testing of contaminants and adulterants in dietary supplements, a risk-based approach. And I will cover a bit about the program side of things. We'll talk a little bit about kind of why is this coming up now? You know, what's happening in the industry. Talk a little bit about how to examine your testing program, how to use a risk-based approach to determine what type of testing you're going to do and kind of how to set specifications, things like that. We're going to deal with this on a high level. So with the 45 minutes or so that we have today, of course, we're only going to scratch the surface a little bit, but certainly ask questions in the chat as we move along, and we'll try to answer those as best we can at the end. You know, obviously using suppliers, we'll talk a little bit about the risks involved in that and how you should qualify those suppliers and deal with that on an ongoing basis. And then Lukas will deal with the analytical side, talk about the validation. How do we look at approaching contaminants and adulterants and setting up methods, you know, the detection modes of that. So he'll talk a bit about the more scientific side of things when we get into that, and some of the current methods that are out there that you may be looking at or may be interested in understanding how we deal with those. Just a little bit about what we see today and what kind of spurned on this. We were talking internally a little bit that we've seen a lot of activity in the contaminant and the adulterant space, especially as a laboratory. We get a lot of inquiries. So it made us kind of look at, you know, what's kind of happening in the industry, or the things going on that are making our customers look a little more closely at this. And when you start to look at the headlines and certain things that are in the, you know, on the media, there certainly is some activity going on right now. And I think part of it is because, you know, COVID has kind of allowed now FDA to start to look a bit more into some of these things, audit more facilities, getting more audits out there. So there is some headlines here. Of course, tainted dietary supplements are in the market today, and Lukas will talk a bit about that in his presentation. I'll touch on it briefly as well. Certainly the weight loss, sports nutrition, sexual enhancement categories are a hot topic with Amazon. You know, if you're selling through Amazon, you're probably aware of some of the analytical testing that has been required of you on that. So we'll talk a bit about that as well. But there's a lot going on in the industry. And again, I believe the dietary supplement industry has done a tremendous job of really, you know, putting safe products in the market. I certainly don't want to paint the picture that all the, you know, all the dietary supplements out there are bad products. But sometimes, you know, in the media, you see these things happen. It kind of paints a picture, you know, one bad apple kind of a scenario. So, but all in all, I feel like the industry does a good job. And, you know, we are what I would call a self-regulated industry in many ways. You know, we rely on, you know, the brand owners are responsible for quality and safety of the products. So obviously, you know, all of you on the call today are interested in testing and doing the right things. And generally speaking, most companies are doing the right things that want to do the right things. And we're going to touch a bit about how you may do that, and especially on the limited budgets that might be out there as well. Obviously, quality, begins at the beginning. The GMPs pretty much specify that. They build a kind of a ground up approach to this. So, part of it is these first three buckets: ingredient qualification, in process, processing, blending, things like that, and the finished product phase where contaminants and adulterants can be introduced. So there's a lot of responsibility at those stages to try to control and mitigate and address contamination and safety and adulteration as you move along. So we'll talk a bit about how to look at those things, and again, with the reliance on suppliers and third-party manufacturers and third-party laboratories, it is really incumbent on you to do the right thing, to audit these facilities, make sure they're working on your behalf and doing the best they can to make sure that we have safe, quality products in the marketplace. One big area, I would say, and this has been around for a long, long time, probably since the beginning of the GMPs about 15 years ago, is setting meaningful specifications. Especially where it is critical to quality and with a focus on safety, which is, I believe, where FDA focuses a lot of their attention. I would say they're not as focused on, whether you have a low amount of vitamin C, but certainly things that may pose potential risks to the consumer. Those are the things that they're really looking at. And one place that they really see a lot of warning letters and over the years, which has been on the top of the list, is lacking of specifications. So having a lack of specifications designed for the products. As we know in the dietary supplement industry, identity, purity, strength, composition, those things really stick out. They show up in the GMPs. They're pretty well established as to what those mean. And today we're going to touch, though, on the limits on contaminants. How do you address contaminants or likely contaminants that are in the products and materials that you have? So, not all products create the same risk with the same contaminants. So you may have to take an assessment of each ingredient individually to try to assess what you should do and how you should test those products. And just very recently, I wanted to show a couple of examples of what we're seeing from warning letters out there. This one is very recent, from, I guess, last month now, talking a bit about the weight loss category, as I mentioned earlier, and again, Lukas will touch on it. Sibutramine, a weight loss compound found in dietary supplement, which is obviously not a good thing, a very dangerous thing as well. So FDA is focused on that. So this warning letter really highlights that, that they're seeing this in the market, and obviously without having to go through any pre-approval process or pre-safety assessment type of processes, some of these products do get in the market. And sometimes unbeknownst to the manufacturer, they may not have done the right thing or the proper thing to know that their ingredient was tainted. So there's certain things that you may have to put into play in your thought process as to how you would deal with some of these high-risk ingredients that you're using. And one more, just to highlight the specification piece that I mentioned. Again, this has been probably on the top of the list for the last 15 years, as I mentioned, having a lack of specifications. This is just one from last couple months, that they failed to establish specifications on limits of those contamination that may adulterate or lead to adulteration of the product, such as microbes, pesticides, heavy metals. Again, these are all topics we may touch on. Again, we're not going to talk much about microbiology per se, but obviously heavy metals, pesticides, things like that can infiltrate ingredients that you may have to look at and set specifications for, to be able to understand the quality and safety of your products. So I did mention a couple of the terms here, contamination and adulteration. Some people look at this kind of as a, very similar. There is some distinctions obviously between the two. Contamination is the presence of an unwanted component in the product that may pose risk to safety. Again, these are unintentional things that happen, with the things that infiltrate the ingredient, maybe the cleanliness, and they come in different forms, like chemical contamination, like lead. Pesticides is a chemical that can end up in plant materials. If you're not screening for that, you may want to think about that. The physical contamination, like insect fragments, parts of the manufacturing equipment, things that happen from a physical standpoint that may also contaminate the product. Biological and microbiological pathogens, toxins, really belongs in all specifications designed around products that go to consumers. So understanding the microbial load of the product is a very important piece of understanding, where to set those limits. And then environmental and allergenic. So a lot of ways that in different places that could contaminate products, and a lot of spots where those could come in, especially at the ingredient phase as well. Adulteration contains any ingredient, and I'm going to talk a little bit about intentional adulteration slide later, but the definition that I see for adulteration contains any dietary ingredient that presents a risk to health when used as recommended, contains an NDI that lacks safety standards. So you can see on the screen here, a bit of a difference between contamination and adulteration, but probably more problematic as we see it, and especially to the consumer, are those intentional adulterations, things that happen for very nefarious reasons, for trying to make more money, more profits. Economic adulteration is exactly as it states. It's a less expensive ingredient that generally replaces the more expensive labeled ingredient. So we see this oftentimes in the plant material space. You'll see it among other things. So if you do have, some ingredients that are prone to economic adulteration, you may want to set some limits or do some testing to make sure that, ingredients aren't sliding into your pipeline that may have some of these compounds there. And pharmaceutical adulteration, again, touched on sports nutrition, weight loss, sexual enhancement. We see this as an active drug is placed into a natural product to try to elicit, the response that you need. So those two things we'll touch on a bit as well as we move along. Causes of contamination, pretty straightforward here. I think we all have a good handle on where contamination can be introduced, but generally, in the ingredient itself, where there are impurities and other natural compounds that may be there, that, if you don't have a handle on that in the ingredient phase, it's certainly going to end up in the finished product. So understanding where those impurities lie and where they come from and what they are, especially important to designing a good test plan. Cross contact, leftover residues from manufacturing. So, maybe residual solvents or poor cleaning of the equipment could pose risk to the product as you're blending things through. Poor hygiene, poor cleaning, of course, people have to touch things and handle things. So those are always places where contamination can be introduced. So understanding how to mitigate those sources is very important in the process of designing good quality, safe products. Obviously we're using water and other environmental components. Those can certainly introduce contaminants to the mix. And the physical equipment, fragments, carryover, if things are, obviously good control of the physical equipment is important to also limiting the amount of contaminants that may end up in the materials. So where are the highest risks? I touched a bit on, obviously, at the ingredient phase. And there's a lot of, and I would say these three phases in the ingredient, the supplier and the manufacturer all add a level of risk that is really incumbent on the brand owner to understand kind of where those are, and to qualify these folks that you're using to make sure they're doing the right things on your behalf. In the ingredient, obviously the geography of where things are coming from has a big impact of what risk might be there. The ingredient type, how it's processed, very important to get a handle on the processing of the ingredient is where some risk can of course come in. The storage and handling, are we introducing any risk when we store the products and for later use? Identification, authenticity, are you using the proper or the authentic material? Really what you're purchasing should have the identification check. So those are places where risk can be introduced, and the intended use of the ingredient. The supplier, of course, we all rely on suppliers. We do as a laboratory, you do as perhaps a manufacturer, a brand owner. So making sure that the supplier has the proper experience and understanding, a handle on the regulatory space. Make sure the sourcing of the materials is up to par for the quality of the ingredient that you're looking for. So having a handle on your supplier and really keeping a good relationship with those suppliers is paramount to creating good quality products. Last but not least, the manufacturer. The facility has a big impact. If you're changing facilities, you're going from one manufacturer to the other. There's a lot to be done to qualify and to try to make sure things are being done correctly when you do that. The audit history, understanding how the manufacturer has fared over time during audits, and you should be doing audits of all these under all these circumstances to make sure that they meet your quality standards. Sourcing of materials, process changes, all of the above really are where risks can get introduced. So having a good relationship and a good qualification process for these people that you're dealing with on a regular basis is very important. Again, mitigating risk, qualify the suppliers. So have a good qualification system, questionnaires, in-person audits. We, of course, as a laboratory, we have customers come in auditing our processes. I suggest you do the same thing for the manufacturer, for the laboratory, for the ingredient suppliers. Make sure that you have a good system for qualifying those suppliers, so when changes are made and things happen, you have that all fully documented. And you'll be relying on certificates of analysis from your ingredient supplier especially. So how do you look at that and, without having to test everything all the time? We all know we have budgets. How do you qualify the C of A's you're getting? There is some resources around how to do that, how to deal with that. So it's very important to know that, how to qualify a C of A. And then once you've established a good quality C of A, you've verified it, and you're confident that time in and time out that C of A is going to be, or that material is going to be the same, you can reduce your testing to some degree. So understanding the risk of that as well. So relying on the suppliers, trust but verify. I think it's really where the term comes in. Trusting your supplier, getting to a point where you trust them, but also keep them honest. Verify intermittently throughout time to make sure that things haven't changed, they haven't gone off the rails, so to speak. So there are things that you would do along the way to verify that the quality of the material, the safety of the material is still intact over time. And generally, part of this is done on an ingredient risk assessment. So, if you have 100 ingredients that you use in your facility, probably have a risk assessment for all of these ingredients to make sure that you know what the ingredient is, how it's used, what it's for, is it an active, things like that. I'll talk a little bit more about that in the next slide or two. But having a risk assessment on each ingredient as you use it, so you can, make qualifications on those ingredients as well. And talked a bit about risk, a risk-based approach using risk management principles to create a well-designed qualification program. So what are the risk management principles? Pretty straightforward, I would think. Obviously, again, we all have limited budgets. So how do we use a risk-based approach to, to do the things that are of highest priority, that create the most risk? So, what I would suggest is looking through the ingredients, the products, and assess the degree of the risk. Is it a low risk? Is it a medium risk? Is it a high risk? And you'll have to decide for yourself what that really means to your company, and then determine the type of risk. Generally a safety risk is a high degree of risk. So a lot of the safety components would have a high degree of risk. So you'd probably want to do more testing, more evaluation on those types of materials than you would a low risk product. Makes a lot of sense. Is it a quality risk? Is it a regulatory risk, or is it a service risk? Something that might pose an issue to the service of you being able to provide the services to your customers. So those come into play when you're looking at determining what type of risk it is. So assigning perhaps a number, a scale of 1 from 1 to 10 or 1 to 5 on a risk-based scale is something you may want to do, so you can then prioritize later on. And the risk factors, is it the supplier, the component, the manufacturing? I talked a bit about this in an earlier slide. So understanding what factors come into play that pose risk to the the quality of your products. An ingredient risk assessment should encompass a lot of these different aspects. So determine the type of the ingredient. So what's the intended use? Is it an active ingredient? Is it an excipient? So once you've determined that, they may have a different risk level that you might assign to those. Understand the pipeline. How many products is this going to? What's the end user? Who's the source and supplier? The country of origin could pose a risk. So documenting all this is very important in trying to understand where those risks happen, and understanding regulatory limits where the product is marketed. You might be selling products in the US today, but you may want to sell them in Canada or overseas. So making sure you understand the regulatory requirements in all of those regions is an important piece to understand risk and know where to assign specifications and those types of things. And likely contaminants, I talked a bit about that. The different types of component will dictate what components, what contaminants you may have to assign to those as well. So components that are prone to adulteration. This is just certainly not an all-inclusive list, just things that I've seen. Oils are prone to adulteration. Certain oils can be cut with lower grade oils. They all have fatty acids and compounds that can't be distinguished from one another depending on the oil sources. So some of those are prone to adulteration. Proteins, of course, you probably remember back in the day when melamine was added to infant formula in that industry back about 10 years ago now, I think, to add nitrogen to bolster the protein content and kind of fool the testing techniques that were used for protein. So if you're using a lot of proteins and things like that, you may have to establish a means of eliminating some of those non-protein nitrogen sources that may be there. Of course, weight loss and sexual enhancement, bodybuilding, as we discussed, and plants and botanicals and herbal extracts. These are very popular these days. There's a lot of plant materials, botanical materials in the market, a lot of extracts, different plant parts that are being used. They're in different blends and different quantities. And there are known imposters out there that try to, that folks try to pass off as the real thing, that really are a different subspecies or a different variety. So understanding where those risks are, what are those plants, how do you establish, a good program around some of those maybe high risk components. And that being said, here is some of those, just a few of these examples. Turmeric, you know, people add yellow dyes to change the color. Turmeric is a very popular plant material that's used, so it's a high risk of adulteration. So you may want to, you know, look at, if you're using turmeric, you know, make sure you have a good program to make sure you have the proper identification and adulteration tests along the way. Saw palmetto, you know, saw palmetto is one of those plants that creates oils and things that are highly adulterated in the marketplace. So cordyceps, you know, in the mushroom space, we're going to see this probably more and more in months and years to come, where mushrooms are very popular these days, and you're going to see probably some of this going on where there's high risk of adulteration and maybe lack of identity standards out there. So just things to think about if you're using a lot of plant-based materials, grapeseed extract, black cohosh. And these, you know, APA, American Herbal Products Association, has a nice list of things that, you know, that go beyond what I show you here on the screen that you could look at to see if you're using any of these compounds and what the risks are. And one other source of possible risk is the laboratory. You know, urofins, obviously being in the laboratory space, we try to make sure we have all the proper accreditations and validations together. You know, obviously we have, you know, things that we're still shoring up as well. So it's one of those things that, you know, all laboratories day in and day out have to consider, you know, making sure that they have the proper documentation on different matrices and different compounds. So using laboratories that have proper experience and expertise is very important. And relevant targets, this is going to be kind of my pass off to Lukas here in a moment. Again, relevant targets, if you're looking at individual categories of things, pesticides, heavy metals, toxins, residual solvents, plant toxins, environmental contaminants, and the list goes on. Lukas will touch base on a few of these things. You know, obviously we don't have enough time to cover everything, but I'll pass this along to my colleague Lukas for him to talk a bit about his about the technical side of things and detection and method validation. Lukas, when you're ready.
Thank you, Jeff. Excellent overview. I learned a lot too. Okay, so in the second part of this webinar, I'll briefly touch on, let's say, gold standard techniques and trends in the analysis of contaminants and adulterants, and I'll provide an overview of the method validation process that needs to be performed prior to using the methods in routine testing. That's a very important part. And finally, we'll take a look at several selected groups of analytes that, you know, recently are of higher concern, I would say. And I'll provide some detailed information on why these should be tested in dietary supplements and ingredients, and how this testing is performed, at least in our laboratories. So when it comes to the testing of when it comes to the testing of contaminants and adulterants in dietary supplements and ingredients, you know, we need to understand that there are some major challenges the analytical laboratory faces. So typically, especially in case of botanical materials, these are very complex matrices, and we are trying to analyze relatively low or even trace target analyte concentration. And this definitely requires use of state-of-the-art techniques. And in this testing arena, you know, I would say that the major technique used is mass spectrometry and hyphenated techniques. So we are looking at, depending on the properties of the target analytes, at liquid chromatography, mass spectrometry, or gas chromatography with mass spectrometry. You know, in case of elements, we are we are using inductively coupled plasma mass spectrometry and sometimes also some hyphenated techniques. So the main advantage of mass spectrometry over the more conventional approaches, I would say, is really high sensitivity, high selectivity. It allows you to provide or get information on the structure of the analyte, so you can basically use this technique in both qualitative and quantitative way. And depending on the particular type of mass spec, you would use that instrument for different tasks. When it comes to trends, definitely there's a move towards multi-analyte methods when we are trying to generate data for as many compounds as possible. Typical example would be pesticide residue analysis when we are looking at up to 500 compounds that can be analyzed in a single run. And moving forward, we also can use mass spectrometry to look even at compounds that we are not targeting at the time of the analysis. This technique allows us to retrospectively assess the data and look for unexpected or non-targeted compounds. So let's move forward with method validation. Method validation represents key activity in the chemical analysis that is really indispensable for obtaining reliable results and demonstrating that the analytical method is suitable for its intended use. And it's also needed to gain ISO accreditation, ISO 17025 accreditation for a particular assay and obtain formal recognition of technical competence to run the particular test. Here we can see a list, I would say abbreviated list of the most important method performance characteristics that are evaluated in the method validation process. We do need to look at selectivity, LOQ, linearity, and range. Very important for quantitative assays are accuracy and precision, also robustness and stability of both reference standards, chemicals, and samples themselves. The validation process always starts with I would say, gathering of all the information needed in order to gather all the information on what kind of analytes are needed, what kind of levels are relevant to the testing. And all these inputs need to be considered in the method development. Then we run, when our method is developed, we do run some set of pre-validation experiments just to confirm that we can expect good performance on the method, and then we also draft the SOP. Really important piece is to have a good validation plan in place that, again, summarizes all the matrices to be included, analytes, and also materials and acceptance criteria. What is basically acceptable for our method when it comes to its performance. So once we generate the validation data, this data will give us really valuable information on the acceptance criteria that we can further use in routine analysis, including system suitability criteria that relates to the performance of the system. Then we can basically come up with a final SOP. So let's take a look in more detail at respective performance characteristics. Selectivity is of paramount importance. This is basically the ability of the method to determine your analyte in the presence of matrix or other components that are co-isolated from the sample during the sample preparation process. So in this particular case, you can see 3 steroid compounds, one of which, number 3, is actually an ingredient of the of allowed or permitted ingredient in the dietary supplements, while the other 2 are considered pharmaceutical adulterants. So as you can see, it is really important to ensure that you are able to chromatographically or in another way separate signals of these compounds, because otherwise you would be generating false positive results. Next, linearity and range linearity or calibration function in general is really important for quantitation assays. So we do need to make sure that we are able to get good linear response, over the evaluated range. And then range basically gives you an interval between the lowest and the highest concentration that the method is able to determine with acceptable accuracy and precision. Accuracy and precision. So these parameters tell you how close is the value that you determine with your method to the actual true value, the actual concentration of the analyte in the sample. The best way to evaluate this is to analyze certified reference materials that have certified values for particular analytes, or some materials, QC materials with assigned concentrations that may be coming from proficiency testing that are generated from a number of different labs using different methods. The problem is that these are not readily available for all the analytes and matrices of your interest. So if that's not available, you need to rely on fortification experiments in which you spike your analytes into matrix, ideally matrix that's aligned with your matrix scope of the method and which is free of your target analytes, and then you can assess recovery of these analytes. And you would run these samples in multiple replicates under different conditions, and this way you can also assess the precision, which is typically expressed as relative standard deviation. So this table provides just an example of acceptance criteria. I took these from the AOAC International Validation Guidelines for dietary supplements, and as you can see, the lower the concentration, or the higher the concentration, the acceptance criteria get more strict, because your method is basically expected to be more accurate and precise as the concentration of the analytes increases. Limit of quantification and detection, really important. Performance characteristics. There are different ways to determine these, but the approach we take in our laboratory, we typically determine mainly LOQ, and we set our LOQ at concentrations that were shown to provide, at which method was shown to provide acceptable results. So this way you basically show that at your reporting limit you are generating valid data. Robustness, it's an ability of the method to not to be affected by some small changes to the method parameters or the procedure itself, and because in real analysis it may be that your mobile phase composition or your let's say sample preparation workflow may be performed slightly different than per the SOP when it comes to volumes, etc. So you need to make sure that your method can still generate acceptable results when these small modifications are introduced. And of course, stability of reference standards, because you're basing or you're determining the concentration of your analytes using reference standards. You need to make sure these are stable over a defined period of time and under certain conditions. For example, in freezer or fridge, or on the instrument, maybe in the instrument auto sampler and so on. Sample extracts, that's again important to determine because it may be that your run fails and you still want to use those extracts and reinject and reanalyze those. So you need to have data to show that these are still stable and your analytes of interest are not degrading, for example. And same applies to reagents and other solutions that are used to carry out the protocol. So the outcome of the validation, you can set some system suitability criteria for your instrument just to make sure that it performs adequately just prior to starting running the sample. So this can be based on different parameters, including the response of your analyte, stability of retention times, or the quality of chromatographic peaks you are getting on that particular day, etc. So again, system suitability criteria are set based on information gained during development, pre-validation, and validation itself. And on top of making sure that the instrument is performing adequately, you also want to run some quality control samples with each of your analytical run, just to have additional confidence in the generated data. So you can run spiked samples, you can fortify the test samples themselves to prove the method works as expected. You can run CRMs, QC materials, and also blanks, which are important just to demonstrate that there's no carryover or cross-contamination of your samples during the sample prep or analysis itself. And you can also run some calibration verification solutions which can be, for example, standards that were obtained from a different source than that used for your calibration standards you use for quantification. So all these are really important to generate routine, high-quality data. Just a small note on quantification. Mass spectrometry, brings a lot of advantages. One of the problems is that signal of the analyte, when being analyzed by mass spectrometric methods, is influenced by the co-extracted matrix, and it can either suppress or enhance the response of your analytes. And the issue is that in that case you cannot use solvent calibration standards, so your reference standards prepared in solvent. And there are different ways to work around this. You can either compensate for the matrix effects, so you can, for example, prepare your calibration standards in the matrix you are actually analyzing, which is free of analytes. You can use internal standards to normalize your analyte's response, or use so-called method of standard additions, which is laborious but compensates very effectively for both matrix effects and also any losses of analytes during the extraction process and so on. So this is like the highest tier of quality data, as long as your data are passing certain criteria. And more difficult but still possible way is to improve your extraction. So you co-extract less matrix, also by improving sample cleanup. You can optimize your conditions in the method, so you can maybe get rid of some of the matrix effects. And a very powerful approach is also dilution of the sample extract. So you are basically reducing the concentration of your target analyte in the matrix, but at the same time you are diluting out the matrix. This approach can be used especially when we are employing really modern techniques that are highly sensitive. So let's switch gears now and take a closer look at selected contaminants. I will take a closer look at heavy metals, plant toxins, especially or specifically pyrolysidine alkaloids, and also pharmaceutical adulterants. When it comes to heavy metals, arsenic, cadmium, lead, and mercury are typically of highest concern due to their high toxicity. These and other heavy metals are taken up from contaminated soil, for example, by plants during cultivation, and metals can also be transferred to finished products if your ingredients, obviously, if they are contaminated. Soil contamination usually comes from industrial sources like irrigation, but also maybe poor quality fertilizers or plant protection products. And of course, as mentioned already by Jeff, materials can also be contaminated during processing. The typical heavy metals analysis workflow includes thermal decomposition of the organic components of the sample with the use of inorganic acids and other reagents. This process is also known as the mineralization, and the extract are subsequently dissolved and analyzed by ICP-MS. This approach allows determination of total concentration of individual heavy metals down to low ppb levels, even in very highly complex matrices. In addition to heavy metal analysis or total heavy metal analysis, determination of respective species of metals that may be present in the samples is of high concern, because there may be significant differences in their toxicities. Good example are the inorganic species of arsenic. Arsenite and arsenate are highly toxic, while organic species are not considered toxic. So so-called speciations methods allow determination of both inorganic species along with common organic species. And with our method, we are able to achieve LOQs around 10 ppb or micrograms per kilogram. And this is achieved by chromatographic separation of different species via chromatography or ion chromatography, and subsequent detection of eluted species by ICP-MS. The other speciation assays we are actually currently working on is for chromium speciation, where the goal is side-by-side determination of trivalent and hexavalent chromium that again differ significantly in toxicity. This represents a challenge due to some interconversions between the species during both sample preparation and instrumental analysis. And again, the technique used for this testing is ion chromatography coupled to ICP-MS. So stay tuned. This is something we plan on offering relatively soon. Another group of important contaminants that gained a lot of attention are pyrrolizidine alkaloids. There are over 500 different pyrrolizidine alkaloids and their N-oxides that are known to date. And these natural toxins represent secondary metabolites produced by a number of plant species, basically as defense chemicals against herbivores. So pyrrolizidine alkaloids were shown to be hepatotoxic and genotoxic, and their toxicity may significantly differ between various subclasses and is associated with respective chemical structures of these compounds. Contamination of dietary supplements and ingredients with PAs is typically associated with co-harvesting of alkaloid-containing weeds or accidental inclusion of such plant materials in the herbal ingredients. Alternatively, cross-contamination of crops that do not produce PAs in the field can take place via uptake of these chemicals from soil, from both rotting and living donor plants. It should be said that reduction or elimination of the presence of pyrrolizidine alkaloids in herbal ingredients and finished products is a challenging task that requires integration of some complex mitigation measures at both cultivation, harvesting stage, and also during processing of the raw materials. So why there's a lot of interest into these? On July 1st, there was introduction of new European Union maximum limits for lower bound sum of 21 pyrrolizidine alkaloids and their N-oxides, and these basically also include additional alkaloids that may analytically interfere with these analytes. So basically, they are so similar that they collude under typical LC-MS conditions. The regulated matrices include dietary supplements, infusions, teas, herbs, and spices. And these are the matrices that were shown to be the highest risk when it comes to contamination with pyrazidine alkaloids. This was based on a large monitoring study performed in the EU by respective member states. The issue of pyrazidine alkaloids contamination has been recognized by United States Pharmacopoeia as well. Currently, there's a draft, a general chapter available that summarizes important information on these topics, toxins, including recommended target analytes and also some analytical methods that can be used along with challenges. European Pharmacopoeia published a chapter that actually provides an example method based on LCMS-MS for relevant pyrazidine alkaloids. And finally, there, as a part of the botanical ingredients and dietary supplements integrity program, AOAC has initiated a working group that is supposed to develop so-called standard method performance requirements for test methods that could be used in relevant matrices. So I'm actually a part of this effort, and we are currently close to publishing the SMPR for public comments. Basically, SMPR document is then used as a guidance and set of acceptance criteria for method developers or any candidate methods that would be submitted for official method of analysis considerations. This is an overview or just some basic information on the testing methods we use in our laboratories. We are able to hit the required levels listed in the EU regulation. We are using LCMS. This is just an example of some reporting limits we are able to achieve for the sum of relevant PAs. So when it comes to pharmaceutical adulterants, Jeff already summarized a lot. So obviously it's a profit-driven, fraudulent practice. And here I just want to show the three categories of supplements that are the highest risk. So sport supplements, weight loss supplements, and also supplements that are advertised for sexual enhancement. And depending on the category, there are different classes of compounds that can be used to intensify the claimed biological effect. Currently, we are offering two different methods that allow us to test for a number of PDE5 inhibitors, which are sexual enhancement drugs that were used to treat erectile dysfunction. And on top of those approved drugs, we also cover a number of analogues that have been developed as generics, or are some side structures that were not evaluated in studies when assessing these analytes. And also we are covering a method, or we are offering a method that covers a number of weight loss adulterants, so anorectics, stimulants, laxatives, diuretics, all these kind of compounds. So we are using LCMS again, but this time around with high resolution mass spectrometry, which allows us to look for both targeted but also novel analogues. The approach we use to look for novel analogues is based on structure similarities between known compounds and analogues when they are basically slightly modified molecules of the parent compounds. And the screening or this approach is based on looking for similarities. We are able to basically fragment the molecule, and since parent compounds and analogues share structure, we are able to obtain some characteristic signals we may look for during our screening procedure. So, just to underline the importance of this testing, the Amazon, which you're obviously familiar with, they do require that annual testing is performed for a subset or limited number of pharmaceutical adulterants in high-risk dietary supplements. And again, as you see, depending on the type of product, we see different compounds: PDE5 inhibitors, anorectics, anabolic steroids, SARMs, which are selective androgen receptor modulators. Again, basically muscle growth agents. And we developed separate methods applicable to various types of dietary supplements, and we are able to achieve or basically report results that at levels that are relevant to economic adulteration or adulteration with pharmaceutical ingredients. So now I will ask Jeff to summarize and provide some takeaways from this webinar.
Thank you, Lukas. Great summary of the technologies and the method validation process that we go through. So just to conclude here, and I know we're running up on the hour, so anyways, obviously we talked a bit about, utilizing a risk-based approach to understanding how to address contaminants and adulterants. Kind of look at it from an ingredient standpoint is a very important part of the process. Making sure you have good qualification of your suppliers that you're using, because that's obviously an important piece of the quality of your product. Lukas did a great summary on some of the methodologies that are current. How do we deal with heavy metals, pyrrolizidine alkaloids, the pharmaceutical adulterants. So hopefully we've provided you a bit of insight, and of course, as we move along, we're going to be doing a lot of these webinars over the next several months to cover some of these hot topics. So at this point, Ellie, I'm completed with the conclusion here. I don't know if we have time for any questions, but thanks everybody for at least listening to what we had to say.
Great. Thank you for that presentation. We will do one quick question, and then if your question was not addressed that you asked during the presentation, we will follow up with you after the webinar. So one quick question is, when you say trust but verify, is there any guidance on how you would qualify a supplier's COA for contaminants and adulterants?
Thanks, Ellie. I guess I'll address this piece. So yeah, I mean, the trust but verify piece is obviously, again, you're utilizing suppliers to provide you ingredients that have C of A's, that essentially dictate the quality of the product they're selling to you. But there is a verification step to make sure that what you receive is of high quality. I look to, and again, I think this reference is a good one. You may want to, I didn't put this in a slide, but you may want to write this down. It's the Standardized Information on Dietary Ingredients working group. They did a voluntary guideline on component supplier qualification. It's an excellent document in referencing a risk-based approach. Again, it goes into risk, how you would verify a C of A from a supplier. So if you have a low risk component, it gives you some examples on the frequency at which you should test that component to then be able to reduce the amount of testing you do later on. And then it goes into the medium risk and the high risk. So it's a very good, and it was developed by industry stakeholders. It was a big collaboration. I think this has been around for, 10 years now, maybe, and maybe not as known. So just pointing that out, I like to refer to that with customers wanting to understand how frequently they need to test this. How do you qualify an ingredient? So I would say look that up, and that'd be a great first place to start.
Okay, that will wrap up our Q&A session. I want to remind everyone that a recording of this webinar, along with a copy of the slides, will be available in 3 business days. Thank you for your expertise and presentation today, and thank you everyone for attending.
Thank you.
Thanks, everybody.